and resolution is one of the limitation. We are getting low resolution because normally we are using, and creating a mapping population itself is difficult for many crops. For example, you take perennial crop, it's very difficult to develop a mapping population. These are the limitation which limits the mapping of the genes in particular species and at higher resolution. That's why scientists came up with the other approach called as association mapping, and today we are going to discuss about it. Any doubts about QTL mapping and mapping from till today? And association mapping, it is easy if you understand, like if you just read on surface thing. But if you want to understand in a very deeper way, you should have a brief knowledge about population genetics. Okay, but I'll try to convey somewhat information about what are the basics you need to know about association mapping. But my friend will deal in detail about how to do the analysis. Okay. Then we discussed about what is mapping, just it is determining the position or elements within the genome with respect to identifiable landmarks. Just identifying some landmark, okay, here our gene is there, you go to this landmark, okay, you'll find the gene. Like this way, we are just, this is only called we are mapping. There are different types we discussed: genetic mapping, physical mapping, cytogenetic mapping, and in physical mapping only restriction mapping will come, and comparative mapping. Like normally nowadays we are using comparative, but it's not exclusive class. It comes under linkage or any other mapping method. Meeting the people. Okay, then we'll discuss what is the difference between family-based mapping and population. In family-based mapping, what we will going to do? We will select the parents, we will develop the mapping population, we will genotype and we will phenotype, and we will identify the QTL. But here you have to create the mapping population. This is one of the difficult thing in some crops. And even though you created a mapping population, we are allowing the recombination between the two individuals. Two individuals. But they are differing in only some particular trait. For example, max to max in rice, we will get only 20% polymorphism. Within that only the allele will differ. And we can map the genes which are differ between the parents. But the genes which are same between the parents, we are unable to map. That is one of the disadvantage in the family-based map. And in GWAS is one of the approach. The population-based mapping is approach is called as genome-wide association study, and there are another type called as candidate gene approach. Here we are using a random population. We will discuss in detail, and we will map the genes. Instead of creating the mapping population, we just use the random population. Like take one some 20 genotypic panel, then sequence it and phenotype it, and you will get MTS, marker trait association. Like this, without need of creating mapping population, we are mapping the gene, that is called as association mapping. And it is linkage based, it's not linkage based. We'll discuss in detail after discussing about association mapping. Why family basing is not family based mapping is not successful for some condition. For example, if there is complex traits, for example, you take yield itself a complex trait. It's governed by many minor genes. And to map this many minor genes, like we need a very high resolution map. To high resolution map, we need larger population. It's laborious to do. That's why to map complex traits, somewhat QTL is not that much useful. That's why we need some other approach that is called as association mapping. Then common variants. And in the, like for example, common variant, for example, oh sorry, it is actually, it can able to, you can map rare alleles and common alleles also. But in association mapping, we can't map rare alleles, that I will discuss. Sorry, I included points here. Then genetic heterogeneity. Sometimes due to genetic heterogeneity, we can't able to map because the so much heterogeneity in the variants, there will be, we assume whenever do the phenotypic analysis, we should have the homogeneity of the variants. But many times we will not have that assumptions. That is one of the problem. One allele which may, which two parent, we can able to detect, for example, there will be capital A and small a. For example, the parents will differ with these two alleles. We can able to map only these alleles. But sometimes capital B, and here also capital B, capital B. We can't map the gene B. The alleles which are differing with the two parents, then only we can able to map, because they will recombine and we will get a linkage map. But if they are same means we can't able to map. That is one of the problem, because in mapping population is just a product of the recombination between the genes which are different between the parents. If the trait is not different between the parents, we can't do a mapping in this thing. That's why it can only the alleles which are uh, differ between the two parents, that can be detected, but which not differ between the two parents, that cannot be detected in the family based mapping. And low genetic map resolution of the genetic map is very low because of limited recombination. You can see this diagram so that you will get very better understanding. See, for example, this is a case of family based mapping, this is a case of association mapping. In family based, we are crossing two individuals. See the recombination, they are not that much high resolution. But you can see this figure. Due to many generations, because of the random population, they evolve during evolution. They normally go recombination between them, and the new genotypes are created because of the recombination only. But they are during the evolution, they have the recombinations were created. That's why you can see we got higher resolution because of the evolutionary recombination events from 1000 to 2000 years. We are tapping this recombinants in the association mapping, but in case of linkage mapping, we creating the linkage mapping. Admitting, why people are admitting so late? I think nobody in the waiting room now. Okay. Then due to we are tapping evolutionary recombinants here, we are getting high resolution. But in case of this family based mapping, what will happen? We're just creating the difference. We're tapping the resources or genes which are differ between the between the two parents. They are only differing. We can able to map the genes which are differing between the two parents. But here we can map all the genes because of the evolutionary recombination. They are different between many genes, and we are using all our individuals. See here, all the individuals are different, and due to the recombination during evolution created this random population, and we are using this random population for mapping so that we can tap more recombinants so that we'll get more resolution in association mapping. That's why we are going for association mapping, to have more resolution map by tapping the more recombinants events occurred in the evolution of the particular plant species. Then, and it is based on the linkage. The family based mapping is based on the linkage between the genes. Either two genes are linked or not, based on that we are mapping. Here, linkage disequilibrium. We will discuss what is linkage disequilibrium. It means a non-random association between the alleles of two or more loci in a population, irrespective either they are linked or unlinked. This definition you should remember. Even though some many things, because of linkage only disequilibrium happens. Yeah, because of linkage disequilibrium happens. But even though the two genes were not linked, means for example, this is a chromosome number 1 and this is the chromosome number 2. If two genes, gene A and gene B, present on two different chromosomes, it will assort independently. General assumption, independently. Assortment. Independently they are assort during segregation. This is the problem. Sorry. We'll wait another 1 minute so everyone can join. For example, if the two genes are present on chromosome A and chromosome B, normally they assert independently during segregation. But even though the two genes are in the different chromosomes, sometimes they will not assert independently. They will not assert independently. They are they are segregate together. For example, A is there, definitely capital B will go. Like that, there will be favorable assortment. Means A always go with B. Means a non-random assortment. Even though they are not present in the same chromosome. Normally, we think genes which are present are, for example, C. A and C, because they are in same chromosome, they definitely they are in linkage and they will assort together. They will not go for independent assort. But the genes which are on chromosome A and other gene in chromosome 2, sorry, chromosome 1 and chromosome 2, they are in different chromosome. Normally, it should assort independently because they are not linked. But sometimes, because of some due to other epistaticus or other hitchhiking effect, we'll discuss what is hitchhiking effect. The genes will assort non-randomly, even though they are present on two different chromosomes. That's why when we define what is linkage disequilibrium means, it's a non-random association between the alleles of two or more loci, irrespective of their physical location. Normally, during linkage, it will be the common phenomenon. They will assert non-randomly. But even though they are wherever it is present in the gene, wherever present in the gene, they will assert dependently. Okay, they are not assert independently. That is called as linkage disequilibrium. Maybe the question existed so many times in the SRF. The questions were asked either the LD between only linked genes or LD may happen unlinked genes also. LD can happen between the linked and unlinked gene also. It's not say that LD only happens in the when there will be a linked genes or LD can only happen there is a only unlinked genes. It's not like that. LD may exist either the genes which are linked or the genes which are unlinked. Maybe due to epistaticus or hitchhiking effect, we will discuss how it can affect. And what is haplotype? We will discuss later. It's a combination of S and P. They inherit together. We will discuss. See, for example, this A and B, they are two different genes having the different alleles. These are the different alleles. For example, this capital A, capital A, capital A, capital A, and small a, small a, small a. There are dominant allele, take recessive allele. This capital A allele either can go with B or it can able to go with small. See, A is going with small b also, A is going with capital B. And here small a is going with capital B and small a is also going with small b. Small b also may happen. Small a can have independence to go with either capital B or a small b. Or a capital A gamete have an independence to go along with the capital B gamete or small b gamete. This is called as an independent assortment, which we discussed. This is called as equilibrium. Means A have can randomly mate with the capital B allele, also can mate randomly with a small b allele. But you can see this situation. Capital A is always going with capital B. See, it's never go with a small b because it's due to linkage or due to linkage disequilibrium. Capital A is always going with a capital B, but it never going with small b. Here small a is always going with small b. It never goes with a capital B. Look at the frequencies. For example, they are all in equal frequency. The assumption is well fitted. Now you can see the genotypic frequency. This is gene frequency and this is genotypic frequency. The probability of capital A capital B is 0.25. Probability of capital A small b is 0.25. Probability of the small a and capital B is 0.25. Probability of small b is 0.25. Means probability of all the genotypes. For example, you take the parents are capital A and small b. Maybe progenies are having A small a, sorry, B, and you'll get equal frequency of the F2. means your recombinants is equal to parent. Recombination frequency, recombination genotypic frequency is equal to parental genotypic frequency. Then that is called as equilibrium situation. Means recombinants is equal to parental gametes, because they will not assert they will assert independently, and they will recombine. See, probability of A and capital A, capital B, and probability of A and small b, and probability of small a and capital B, probability small a, small b. All the genotypes, the possible recombinant genotypes have equal frequency. But you can check here. The probability of capital A, capital B is 0.5, and probability of A small b is 0, because A never go with small b. Probability of A small capital B is 0, because small a is never goes with capital B. But probability of small a small b is 0.5. Here, the P of AB and P of small ab is not equal to recombinants P of A capital B and P of A capital A small b. This situation is called as linkage disequilibrium. Either it may exist, either the genes which are linked or the genes without linkage. Then how we can detect the linkage disequilibrium? There are several methods to estimate linkage disequilibrium, either by D based, D value based, and R square method. There are so many, but D prime is R square is mostly used. For example, how to estimate the D? We just take this formula. For example, here probability of A, like there will be a frequency you will know, and genotypic frequency we are going to know. We check probability of A how much? Probability of A 0.5. We write 0.5 into probability of small a small b 0.25. Sorry, here 0.25. 0.5. Then minus probability of capital A is 0.5 into probability of A and B is 0.25. Just check what is the answer. If it is equal to 0, then there will be, what is the answer? Okay. I'm audible.
Yes sir, yes sir.
0 होगा sir.
Then the population will be in, if a D value we are getting 0, the population will be in equilibrium. Means P square plus 2PQ plus Q square is equal to 1. Means the population is in equilibrium. When population will in equilibrium? Can anyone tell? I hope you all know very well about Hardy-Weinberg.
When there are no evolutionary forces like selection, migration, mutation.
You're directly going that. What is the definition of equilibrium population?
Random mating is there, and gene and genotype frequency are equal throughout the generation.
Yeah, when a large Mendelian population, the gene and genotypic frequencies are equal across the generations. Okay, this is called as Hardy-Weinberg equilibrium. Means P square plus 2PQ plus Q square is equal to 1. If it is not equal to 1, then the population will be in disequilibrium. And here, because of, and the condition exists when there is a random mating of an allele. This is the first assumption you should have. The population you should tell in the equilibrium, there should be random mating of random union of the gametes. Random union I explained means any allele of the particular gene can able to recombine with any allele of the other gene. See, capital A is recombining with B, capital B, or a small b. This is called as random union. But it is not non-random union, means capital A is always go with B, small a is always go with small b. It's never go with a different combination. It's never a random in nature. That is called as non-randomness. When the gametes are combining non-randomly, there will be a existence of disequilibrium in the population. Means the p square plus 2pq plus q square will never equal to 1. This is the one thing you should remember. Then I think that this formula, the equation, you get to know how to do the D. Then depends on allele frequency. Normally we are not using this. We are more, we have more, like we have another approach called as D prime approach. Means we are getting D value here divided by minimum P of A into P of small b, P of small a into P of capital B. Here the values are there, you can substitute values, you are getting value. When D is more than 1, you use this formula. When D is less than 0, sorry, D is more than 0, you use this formula. When D is greater than 0, use this formula. And when D is less than 0, use this formula. Okay. Then another approach is called as R square. It's most often used, these two methods, D square and R square, to estimate the LD. You just square the D value. From this formula you are getting D value, and you square that and divide it by probability after capital A, probability of small a, probability of capital B, probability of small b. See, D square is, we are getting some from that you just check non-random. Here 0.5 P of D square, for this another, how much you are going to get? P of A is 0.5 and P of small a small b you are getting 0.5 minus P of A is 0.5 but into 0. Then 0.5 into 0.5, how much you are going to get? D 0.25 you are going to get. Means 0.25 whole square divided by P of A, 0.5 into 0.5 into 0.5 into 0.5. You will get R square. This is your LD in the population. Okay, understood how to estimate the LD? And this method, you don't worry, nobody is using that methods. Okay. Now you understood what is the linkage disequilibrium, how to estimate the linkage disequilibrium. For example, this much is more than enough. In this year also they were asked simple question as LD. LD may exist between the linked or unlinked genes, and LD, the definition of LD, non-random association of the alleles. This year also they asked the same question. Okay. Then what is the factors which affecting the linkage disequilibrium? Means when the chances of LD is very high, now you can tell the evolutionary forces, all those things. Can you repeat? When the population will not going to be under equilibrium. For equilibrium, what are the things we need to maintain? If Hardy-Weinberg law, p square plus 2pq
that
plus,
should be non
when there is no selection,
no mutation,
when there is no migration,
mutation and genetic drift.
no mutation, and there is no genetic drift. I hope you are studying from last PUC days. I will not go into detail. When there is no selection, when there is no migration, when there is no mutation, when there is no genetic drift, the population will be in equilibrium. And there should be a random union of the gametes. And that too in large Mendelian population. If the population will be large, then only there will be random union of the gametes. For example, in a classroom, you having 100 members. You can make friendship with any other member. But in class, you have only 6 members or 10 members. The probability of assorting randomly will get reduced. You can have friendship with only someone. The population size matters. When there is a larger population, LD will high or low.
LD will be low, sir.
Yeah, because there will be, if there is a larger the individuals, the random union will be more. That's why when there is a population size is large, we are getting larger the population, higher, lower will be the LD. Sometimes there will be subgroups in the population. That's another matter we will discuss whenever time, like next coming slides. Then normally there should be, population should be large, then your LD will be less. And Uh, if there is a small population size, there will be higher LD. Because smaller the population, the alleles may go together only. They will not. They don't have chance to mate with randomly with another individual. The population itself is. This is the one of the important factor. Looks very simple. The government investing crores of money into this to maintain the to avoid the inbreeding to increase the sample size. For example, in the wild species, you take tiger. Hardly how much, sir, lion. Tiger or more lion. Hardly how much population is there in lion now? 300, 400, 800. See, this much limited population. There may be a chance of inbreeding. There may be inbreeding depression, maybe the outbreak of the disease. There is a chance of genetic drift because of the population size is smaller. Chance of genetic drift is more. That's why the maintaining the diversity and maintaining the population size, it's very important. So much, even so many population geneticists are working together. Like working, there is separate institute is there to conserve the germplasm, to maintain this. There is separate institute is there. That much importance of this actually this concept, LD concept. That's why when there is a smaller in the population, higher will be your linkage disequilibrium and inbreeding. More will be the inbreeding, more will be the favorable assortment of the gametes, more will be your linkage disequilibrium and very low recombination. See, when recombination is more, definitely there will be dependent assortment. Means the distance will be very high. There will be chance of more recombination. That's why we are going to get a lower LD value. That's why normally for self-pollinated crops, there will be lower recombination because they will self-pollinate. They will never combine with the other individuals. They never cross-pollinate with other individuals. In that case, normally LD value is very, very high for self-pollinated crops because low recombination rate due to higher self-pollination. This point you should remember. Many times it asked in the exam. For example, if the LD is very high between self-pollinated and cross-pollinated, the answer should be LD is very high in the self-pollinated crop when compared to cross-pollinated crops. For example, you take a rice, we have getting like nearly 500 kb of LD we are going to get. This what the LD is very high in the self-pollinated crop. And population admixture we are going to discuss means there is a subpopulation in the within the population there may be a subpopulation means the population the some groups in the for example you take this is the total population there will be subgroups and the individual within this group they never mate with the other group means they never share the alleles between the other group means there will be stoppage of random union. That's why when there is a higher the subgroups in the population there will be higher will be the LD value. This point you should remember. And the epistasis. What is epistasis? One gene will affect the other gene. For example, you are selecting flower color, the governed by gene A. But seed size, it's governed by capital B. You are selecting one flower color, red color flower. When there is a red color flower is there, this red color flower is epistatic to B, means red color grain size. Means whenever you select the red color flower, due to epistatis, red color grain also selected. Due to this unintentional selection of other trait along with your selected trait due to epistasis, this will increases your linkage disequilibrium. And this is also called as an hitchhiking effect. For example, another effect called hitchhiking effect. For example, here capital A, capital B, and capital C is there. They are very tightly linked. But sometimes A is selected, C also selected unintentionally. This is called as hitchhiking effect. You don't want to select this C, but whenever you're selecting the capital A, C is because of these neighboring genes, it also gets selected. This is called as hitchhiking effect. And due to this also LD will be very high. Then when LDV is very low, just you make opposite. When the population size is very large, when there is so much outcrossing, when there is a high recombination rate, when there is a high mutation rate, when there is gene conversion. Gene conversion I will explain. Anybody know what is gene conversion? What is gene conversion? Actually, it's a non-reciprocal exchange of the chromosomal region between the two individuals. Normally, in crossing over, it's a reciprocal exchange. This region it will go here, and this region it will go here. But only this region is coming to this region, but this region will never go to this region. This is called as a non-reciprocal crossing over. Due to that, there will be a gene conversion. For example, there is a and A frequency will be there like 3:1. Due to this gene conversion, it will become 2:2. Means there will be reduction in the capital A, but increase in the capital A. This conversion of capital A into small a due to this non-reciprocal exchange of the chromosomal regions. This is called as gene conversion. It is beautifully explained in the Bidising book. Please read it. Due to this also, there will be a greater reduction in the linkage disequilibrium. Then there will be LD decay. Means if there is a distance between the two genes for increases, there will be linkage also decreases. Due to that, LD also decreases. Means there is a decrease in the linkage disequilibrium value due to increase in the distance. When there is a more genetic distance, there will be decrease in the linkage disequilibrium value. That is called as LDDK. That is called as spatial LDDK. Means when the space between the two genes are increasing, your LD will be decreasing. LD will be decreasing. This is called as a spatial decay. There is another type of decay due to more number of generation. Like for example, F2, there will be some LD value. During F6, the LD will greatly reduced. Like 1 by 2 power n times your LD will go into reduce. Okay, because of why? Because of increase in the homozygotes, decrease in the heterozygotes. Due to the number of recombination events you will going to increase. Definitely, when there is a high recombination, there will be decrease in the LD value. This is called as temporal LD decay. Means the decrease in the LD value due to the more number of generations. That is called as temporal decay. Decrease in the LD value due to higher space between the two genes. The distance is increases, your LD will decrease, means that is called as spatial LD decay. Due to the more number of generation, your LD will be decreased, then it is called as temporal LD decay. Then for each generation, for each random union, there will be one half of the 1 by 2% will going to decrease. For example, in one generation, it will be 0.5. If there is a random union at one generation also, 0.5 divided by into 1 by 2. This, if you have n number of generation, 1 by 2 power n into LD value. LD by 1 by 2 power n will be your LD. It's n is the number of generation or number of the random mating. Random mating will decrease 1 by 2 power n into LD. Where n is the number of times where a random union will occur. For example, 3 times. 3 in 3 generation, your random union will occur. 1 by 2 power 3 into your LD value will be your answer. Like this, we are estimating the LD. Even though if they given okay this number of generation, for example, in generation 1 there will be some LD value. After 3 generation of random union, what will be the LD? Like this, so many questions we're going to ask. You should ready to answer it. Okay. This is what LD and LDDK. Then what is association mapping? Actually, I am very fast because my friend will take in detail. Don't worry. Just I am covering. You should have basic idea before you because maybe he will is expert in that. He may teach in a very in detailed way, and somebody will not going to understand because like if he teaches so analytical way, difficult to understand. That's why I'm giving a brief idea today so that it is easy for understanding this class. Then what is association mapping?
Hello, sir.
Using this LD. Hello. Okay,
Sir, you said na about that in every generation the half to the power n the LD will be decreased.
yeah.
And sir, we also like in our JRF time we used to read that if any means evolutionary forces occurs in population that there will be no equilibrium. But since we remove any equilibrium that evolutionary force in the next generation itself, the equilibrium is restored. So can you
See,
please
that
means
is
tell that
in
case
a
also?
large population,
Yes,
there
sir.
will be a small amount of see max to max how much LD will occurs. If the population, if random union occurs, then definitely it will going to get. But point also, for example, max to max how much LD we are going to get. The frequency is very, very, very less. That's why the population for the it is for one loci. How many loci you are going to have? If you take in that way, in one generation also, population will come to nearly equilibrium. Okay. For one loci I am discussing, but that is in the average of all the genes. When you take the account of all the genes, that will in one random union only, we are getting an equilibrium. But some persistence of LD will exist. It's not like that LD will definitely become zero immediately of the random union. Well, the population come to equilibrium, but some part of the LD may exist. Okay. That we are
Okay, sir.
using for mapping. Okay.
Okay, sir.
And when you take into account of a large number of genes, it will happen. But when we are now discussing for our understanding, we are discussing about particular locus. Then, what is association mapping? What is association mapping? Means by using an LD, we are associating some marker with the trait. That is called as association mapping. Means there is a LD exist between marker and the trait. Now we discussed about the LD exists between the two genes, the gene A and gene B. Think this is one marker and your B is your governing for some trait. We are identifying any marker is having an LD with any important trait. To identifying this process is only called as association mapping. Means we are associating some marker to the trait. How you are going to associate? By the values of LD. Means we know that when they are in LD, they will definitely inherit together. That's why we are checking the marker is having an LD with any important trait. That process is known as association mapping. Then how we will do? For example, first we have to choose a germplasm. Then we have to phenotype as we are doing in our mapping, like QTL mapping. Then we have to genotype same as like this. Instead of using mapping population, we are using a random population. That is the only difference. Then we are estimating an LD value. There we are estimating recombination frequency values. Here we are estimating LD values. The formulas I told, the same formulas they are used for estimating the LD. After estimation, there were several models to associate the trait and the marker. There are several models called as general linear model, structured association model, multi-mixed linear model, and another blink model, forum CPU. There are several models. I'm not going to discuss, but today. But there are several models are there to associate marker with the trait by using the linkage disequilibrium. You genotype the individual with a marker, you phenotype, you combine these two information, estimate the LD, and you finally you are going to get marker which are linked with your trait. This is the only association mapping overall protocol.
Sir, one
Then,
doubt, sir.
Yeah.
Sir, in that scheme, sir, we have to, they have mentioned somewhere that we have to use some background markers to check the population structure, sir, and
Yeah, that is I
relatedness.
will discuss. I will discuss that one. Okay, just see whenever discussing the basic idea, you should think in a basic way. What we will do? We have a population, genotype, phenotype, estimate LD, then associate MTA. I told this much. I'll come by one step. How to genotype, how to phenotype. See, I have 4 to 5 classes for association mapping. But whenever teaching the basic idea, you should follow me, okay? Because you are teaching how this can be happen, you'll forget all the things, and it will confuse for other also. Now you got a general scheme. What we will going to do in association mapping? First, take a large population. Okay, like for example, in my case only, I'm using 144 individuals, like nearly some genotypes, 100 plus genotypes. Then I will phenotyping. Then I will genotyping. Then I will estimate the LD values. Then I will get marker rate association. This is the overall protocol. You should know this protocol first. At least you forget everything. If you remember the simple way, you can assume because question will ask this much only. Many questions will based on this scheme only. They will know nobody will going to ask in detail. Wait. Okay. See, that's why, like you just know this. I will discuss in detail how to analyze the population structure, how to genotype, and what is the why we have to do that. All these things we will discuss. But before going to do anything, you should have a brief idea about what we are going to learn. For example, when you are developing a house, what we will do is just have a blueprint. How pillars should be raised, how gate where gate should come. If you are studying how like in detail, first when you have a blueprint only, you are getting an idea. Okay, I had to reach there, what to do, and what hurdles will come, how to overcome it. That way you have to learn the things. Okay. This here, the general scheme is there is a large population. You had to do phenotyping. You had to do genotyping with the marker. Then you have to estimate the LD. I, I discussed how to estimate the LD. And you know what is LD now? You know what is phenotyping? You know what is genotyping? We discussed in detail in other mapping methods. Same way. Then you are identifying a marker which associated with the trait. How we will do? We'll discuss. Okay, don't worry. Then what is the assumption? Assumptions are based on this. The marker locus is sufficiently close to the trait. Means the marker which are very close to the trait, whenever the trait is going to inherit, the marker also going to inherit. If you choose this marker, you are indirectly choosing the trait. Ultimately, it's used in the marker assisted selection. This is the assumption. Means your marker allele will inherit with your trait allele. Why it is inherit together? Because of an LD. When the marker and the trait, they were in the linkage disequilibrium, they will inherit together. They will inherit together. This is what the assumption for your association mapping. What is the objective? To detect the correlation between the genotypes and the phenotypes. For example, your AA genotype is there. What phenotypes you are going to get? If AA is there, what phenotype you are going to get? This is what we are finally aiming for. For example, what we will get? We are getting capital A 1 SNP is significantly associated with their antioxidant content. Like this, we are going to say as a result. This we want as a final objective. Means this particular allele is significantly associated with the trait, means they are in LD with the trait. Means there is a LD between this allele, marker allele, and their trait allele is very, very high. They will inherit together. Means whenever you choose an allele, there must be the gene allele will be there. That's why it helps in the indirect selection. This is the one of the important main idea you should know before any mapping method. Then, what is the advantage over high resolution? We discussed, and the genetic diversity. We are using a random population. Either you have breeding lines, either exotic and indigenous lines, either use global germplasms, whatever it can be. Population, whatever it can be. Okay, that's why we are tapping a diverse population. When there were the more the diversity, more number of the alleles. We can tap more number of the alleles. For example, in QTL mapping, one having gene A is having two alleles, either A or small a. In parent one allele, in parent two other allele. But the same gene A having multiple allele, A, small a, A1, A2, A3, capital A1, capital A2, capital A3. They have so many multiple alleles for a gene. But these alleles cannot be tapped in the QTL mapping, but it can be tapped in the association mapping. That is one of the greatest advantage of the your association mapping. And as I said, there is no need of a specialized mapping population. Means, for example, you take a mango tree, as I told, to create a real population, it will take a lot of years. Instead of that, there are so many varieties, so many lines of the mangoes were there already. We just take all the individuals and do association mapping, and we can get more high resolution and without creating any mapping population. In that case, it's very useful. Actually, association mapping first came in the humans for identifying the alleles for the genetic disease is majorly used. Whenever you search the paper, most of their animal disease or human disease, the Jiva's approach is mostly used approach. Because you can't make control crosses in the humans, you can't create backcross population, you can't create real population in the humans. What is the option? To map the particular allele which are responsible for the genetic disorders. Then the association mapping have a greatest advantage. Then candidate gene identification. There also we can identify candidate genes. We will discuss. Then advantages of higher the LD means it increases the power. Means, for example, normally in association mapping it's a low power. I will going to discuss. I already discussed, but I will going to discuss. It's fine mapping and transferability. Means higher the LD means they are tightly linked. Means there if you transfer allele, your gene definitely will going to transfer. If they are more the distance, maybe there will be chance of recombination. The reliability of the selection will go wrong. That's why you should have an higher LD for this one. This one I have is. Then
Sir, how increased powers?
what?
Sir, how increased power means should be low power.
I will discuss. I have discussed in previous classes. I will discuss after so many after end. I will discuss. Okay. In last class, I have discussed power and what is power because I have to explain more again. When I were comparing between association mapping and linkage mapping, I will explain. Okay. I think in several so many times this question asked and so many times I explained. Okay, either you're not listening, I don't know. Either you're not getting, I don't know. Either I am not explaining rightly, I don't know. But so many times it's going to ask like what is power and what is resolution. I think anyone tell about this now? Because I explained many number of times, you
Sir,
should answer.
yes,
Other
sir.
person.
Sir,
Explain.
in association mapping, there is a higher chance that we can get a QTL of very small distance, sir. More resolution. Not like in QTL mapping where you can get a larger area as a QTL, sir. Here you can get definite areas as a QTL, but the power of identifying such a QTL is
Maybe
low
I'm
in
going
this
fast.
area.
Okay. You should write it down at least. Okay. You should not
Sir,
write. You should not mean that.
power is in association mapping. We can't tap the rear alleles, but in QTL mapping we can map the rear alleles. That's why in QTL mapping the power is
Yeah,
high. We can tap the rear alleles, but in QTL mapping the number of generation is so low. That's why the resolution is low. But in association mapping the advances because of through millions of years. That's why resolution is high.
yeah, exactly. Whatever the answer you told is very right, very nicely. That's why you should practice more. Okay, these things one time you watch, you will get everything. You just had the things in the slides. It's not like that. Okay, it will not go in like. You should read, you should recap, you should rewrite. Then only you will get the broader idea about this one.
So actually I did not get like how means increased power. That's why.
No, no, no. I'm not telling. Not only for you, sir. Not telling about you. Just whenever you write, like please write all the things which I am telling, because nowhere it will going to talk. I know, because in many essays, they're not going to teach all those things. Okay. Whenever
Sir,
whenever I speak something, should at least try to note the things, at least headlines. Then to search in the web or search in the book, try to remember those things, because it will not covered in any of your classes. I know, because there's some, like we have molecular breeding on course in in minor, I think, in MSc. But the teaching is like, maybe they will not take classes. So many will not take on the course also, because it's an optional course. So many things, but majority of the questions will come from this part of the. Okay, last year also many questions came, but applied level. That's why nobody could able to answer. That's why you should read more in detail. See why power is more in QTL mapping. You're crossing the P1 and P2. You know that they are differing for what trait you know. And the power of identifying, the probability of identifying the QTL per particular trait is very high, because you know that we're making the crosses for that trait, we're creating the recombination for that trait, and you know you have a control over this. And as you told, the rare alleles, because we know that the allele which are between the parents, we are creating them, and we are increasing the frequency of that particular allele by making a control process. Means tapping, we know the probability of getting the QTL for particular trait is very high chance. That's why power of identifying the QTL is very, very high in case of QTL mapping. But here we are using a random population. We are using a random population. As I said, in only one individual, one particular allele is there, but it's never present in any other individual. The frequency of that particular allele is very low. If in this case, we are unable to tap this particular allele, because the frequency is very, very low. And we are using a random population. We are not creating any control population. We are not creating any control population, so that the allele will not going to increase. The frequency of the particular allele will not going to increase. So you are unable to tap this kind of rare alleles present in very low frequency. That's why the probability of identifying or probability of mapping particular these rare alleles which are present in the very low frequency, the chance is very, very less. That's why power is very, very low. But resolution is very high, because we are tapping the evolutionary recombinants. Okay, that's why power is, in case of association mapping, is very low, but resolution is high. In the QTL mapping, the power is very high, the resolution is very low. Now I think you got. I'm stopping here. Any doubts to this? Please write it down. Okay, what are the things I'm speaking here? At least try to make notes. Okay, because when you make the notes, when you see the previous pages, you will get to know like how orderly, because I covered in a very ordered way. It's not like I'm coming and I'm teaching a random topic. No, every chapter is interlinked. If you understand this chapter only, you can understand the next chapter. In that way, I have prepared the lecture series to make easier understanding. So at least make the notes. Okay, written notes is best actually, because slides is there and I'm creating the slides with very much informative. It's not I'm just keeping the images early, as like earlier. because I know these concepts are very difficult. That's why I'm keeping maximum information in this slide. Try to note down.
Sir,
Okay,
many
bye.
doubts are there, sir. Can I ask at the end of the all 5 sessions, sir? The
Yeah,
end?
yeah, okay. You can ask me you can text me in WhatsApp so that I will note it down and I will explain it very quickly. Okay.
Yes.
I had to go dinner and I have some work. Okay, bye. That's why I took classes 7:30. Okay, normally it should be after dinner, but I have some work. New Year is there. Okay, bye. Bye. Good night.
Happy
night,
New
sir.
Year, sir.
Happy New Year. Sorry, I forget. Happy New Year to
Happy New
all.
Year, sir.
Okay.
Happy New Year,
Happy
sir.
New Year.
Happy New Year, sir.
Year will bring the joy and success. Okay, bye. Good night. Good night. Happy New Year to all once again. Bye.
Good night, sir.